Hello, my name is Brian Duff. I'm Technical Lead for the PSC portfolio. Welcome to PSC Foundations Module 1, Stem Cell Categories and Terminology. After this module, you should be able to understand the definition of stem cells and properties of these cells that make them different from other cells. Describe the classifications of stem cells based on potency or developmental origin. And understand what pluripotent stem cells are and differences between embryonic and adult stem cells. This module contains 3 sections. Section 1, what are stem cells? Section 2, what are the different types of stem cells? And Section 3, what are pluripotent stem cells? We'll also have a summary section at the end. Sidebar sections are used to highlight applications and useful additional information related to the curriculum. So Section 1, what are stem cells? Stem cells are unspecialized cells of the human body. One of the fundamental properties of a stem cell is that it does not have any tissue-specific structures that allow it to perform specialized functions. For example, a stem cell cannot work with its neighbors to pump blood through the body like a heart muscle cell, and it cannot carry oxygen molecules through the bloodstream like a red blood cell. However, unspecialized stem cells can give rise to specialized cells, including heart muscle cells, blood cells, or nerve cells. These cells are functionally defined as having the capacity to self-renew and the ability to generate differentiated cells. More explicitly, stem cells are able to divide and can generate daughter cells identical to their mother. This is known as self-renewal. Unlike most somatic cells like muscle cells, blood cells, or nerve cells, which do not normally replicate themselves, stem cells may replicate many times. Stem cells are also able to produce progeny with more restricted potential, i.e., differentiated cells. To be able to divide without losing the stem cell pool for later use, a stem cell is capable of multiplying through division. After each cell division, often one of the 2 daughter cells retains the original stem cell properties. The daughter cell that loses stem cell properties then becomes differentiated, i.e., a specialized cell that produces all of the proteins required for its proper function within a tissue or organ. It can at best divide only a few times, but more often, not at all. This is, for example, the case with at least most brain cells. In conclusion, stem cells are a self-renewing cell population that is multipotent and thus can generate several differentiated cell types. Section 2. What are the different types of stem cells? Stem cells are able to differentiate into any cell of an organism and have the ability of self-renewal. The ways that stem cells are characterized is key in the fundamentals of stem cell biology. Stem cells can be classified by potency or by developmental origin. The potency or potential of different stem cell types refers to their ability to give rise to specialized cells of the body and can be organized as a hierarchy, with decreasing potency as stem cells become more committed along a specific lineage trajectory. The issue of potency may be the most difficult parameter to incorporate into a widely accepted definition of stem cells. Based on the potency, we divide stem cells into totipotent stem cells, pluripotent stem cells, and multipotent stem cells. Stem cells exist both in embryos and adult cells. Based on the developmental origin, we divide stem cells into 2 types: embryonic stem cells and adult stem cells. Embryonic stem cells are derived from a very early embryo, whereas adult stem cells are found in postnatal tissues, not only of the body, but also the umbilical cord at birth. At the top of the stem cell hierarchy sits the totipotent stem cell, which arises from the union of the sperm and egg. The resultant fertilized egg is capable of giving rise to all the cell types of the body, as well as the supportive extra embryonic tissues, including the umbilical cord and placenta. Following a series of well-defined divisions, the totipotent stem cell gives rise to the blastocyst, consisting of a layer of outer cells called the trophoblast, which have committed to producing extra embryonic tissues, as well as an inner cell mass, which specifies to give rise to either the epiblast or the primitive endoderm. The epiblast cells are pluripotent stem cells, which give rise to the fully formed fetus by the end of development. Through a series of differentiation decisions that follow as development progresses, the pluripotent stem cells undergo lineage commitment events and become limited in their potency, giving rise to multipotent stem cells, which have a restricted ability to give rise to specialized cell types within a confined tissue system. For example, hematopoietic stem cells are restricted to give rise to all the cell types of the blood system. These multipotent stem cells then give rise to the specialized cell types of the body, which are considered to be unipotent or terminally differentiated, as they do not have the ability to give rise to other cell types. It is important to note that developmental potency is reduced with each step. When characterizing stem cells based on developmental origin, there are two types: embryonic stem cells and adult stem cells. Embryonic stem cells are derived from a very early embryo just a few days after fertilization of the egg. And adult stem cells are found in postnatal tissues, not only of the body, but also the umbilical cord at birth. A blastocyst is formed after the fusion of sperm and ovum fertilization. Its inner wall is lined with short-lived stem cells, namely embryonic stem cells. Blastocysts are comprised of two distinct cell types: the inner cell mass, or ICM, which develops into epiblasts and induces the development of a fetus, and the trophectoderm, or TE. If the ICM is removed from its normal embryonic environment and cultured under appropriate conditions, the ICM-derived cells can continue to proliferate and replicate themselves indefinitely and still maintain the developmental potential to form any cell type in the body. These pluripotent ICM-derived cells are embryonic stem, or ES cells. Although some controversy still exists about the potency of adult stem cells and their ability to form different cell types, adult stem cells are generally considered to be either multipotent or unipotent. In the adult body in vivo, there are no totipotent or pluripotent stem cells. Section 3. What are pluripotent stem cells? As discussed in former slides, pluripotent stem cells can differentiate or specialize into all cell types of the embryo and adult, including germ cells. However, they are unable to form non-embryonic structures. Pluripotent stem cells are capable of developing the three different germ layers: the ectoderm, the mesoderm, and the endoderm. There are two types of pluripotent stem cells. One, embryonic stem cells, isolated from the inner cell mass, or ICM, of an embryo. And two, induced pluripotent stem cells, which are reprogrammed from adult somatic cells. The inner cell mass can be isolated from the trophectoderm of a blastocyst stage embryo using immunosurgery and transferred to a plastic dish for culture in vitro. These cells are known as embryonic stem cell, or ESC lines. Many types of differentiated cells can be transformed or reprogrammed into pluripotent stem cells when they are transfected with reprogramming factors. These are genes which encode transcription factors that are normally expressed in ES, or embryonic stem cells. These transformed cells have all the properties of ES cells. They are now known as induced pluripotent stem cells, or iPS cells, since pluripotency was artificially induced. There are differences in potency, origin, differentiation potential, and self-renewal potential between pluripotent stem cells and tissue-specific stem cells listed on this table. Based on the potency, pluripotent stem cells are pluripotent, whereas tissue-specific stem cells are multipotent. Pluripotent stem cells are originated from either the inner cell mass of the embryo or reprogrammed from somatic cells. Tissue-specific stem cells are coming from various tissues in the body. Pluripotent stem cells are capable of differentiation into cells of all three germ layers, but tissue-specific stem cells can only differentiate into cells of a specific tissue lineage. Pluripotent stem cells have an indefinite self-renewal capacity in vitro, but tissue-specific stem cells possess a limited self-renewal in culture. So, in summary, stem cells are unspecialized cells capable of self-renewal and differentiation. They can be categorized based on their potency and development. Pluripotent stem cells can differentiate into all cell types in the body except extraembryonic structures. And there are two types of pluripotent stem cells, embryonic stem cells, or ESCs, and induced pluripotent stem cells, or iPSCs. Thank you for your attention.